most animal populations on the planet, whether it's fish, birds, amphibians, reptiles, mammals, plants, insects, you name it. Most things have experienced huge amounts of habitat loss and major pressure from human beings and now climate change issues. And so their populations have dropped. And when those populations drop a lot, they lose the genetic diversity that was so important to their natural evolution and their ability to adapt. So we have fundamentally compromised the future of evolution for many species on this planet. But that's something that these biotechnologies can help. So if we can think about bringing back mutations from an extinct species to help the ecosystem, we can certainly do the same thing by reaching back in time to specimens from endangered species when they were more diverse and bring back their diversity into their living populations. There is no dinosaur DNA left in the world to sequence, so we cannot understand the genome of, say, a Tyrannosaurus rex or a Triceratops. Without being able to understand their genomes, we can't really recreate them and their traits. You know, if someone in the future someday wants to create their own real Jurassic Park, they will be kind of creating those species completely from scratch. Once we understand DNA enough to be able to make whatever DNA sequence we want, we may be able to make organisms that have never been made before. And people are doing this in bacteria, where they're changing bacterial genomes at such scales that they're rewriting kind of the code of their life. And there are some things that we've decided as a community probably shouldn't happen.
Thank you for joining Once Changing the World, India's first Future Tech Meets Sustainability podcast. I'm your host, Eddie Avel, and today I'm delighted and honored to have with me Mr. Ben Novak, the lead scientist and program manager at Revive and Restore, a pioneering non-profit focused on using biotechnology for conservation, genetic rescue, and de-extinction. He's recognized for his work on extracting ancient DNA, kickstarting the Woolly Mammoth Revival Project, which now is being fronted by Colossal Bioscience, and the Great Passenger Pigeon Comeback Project. So, Ben, really appreciate you taking time and being part of the podcast. Now, conservation, DNA, ancient DNA extraction, you've been involved in this since a long time. Will be great if you could give like a background, your works, and what got you interested in biotech conservation.
Yes. Yeah. Yeah, it has been a long time. I've been working with Revive and Restore since 2012. Um, and for a quick, just a correction, because credit is due where credit is due. Um, so yes, Revive and Restore, which started in 2012, did work on mammoth de-extinction for many years before the company Colossal was formed and took on the project. But George Church, who co-founded Colossal, it was George that brought mammoth de-extinction to Revive and Restore. So I didn't help kickstart it, but I helped with the outreach and education.
Thank
But
you
I
for the
wasn't.
clarity.
Yeah, I was not really involved in the science of mammoth de-extinction. But absolutely, passenger pigeon. That's what I lead. I actually came to the job through my early work on passenger pigeon DNA. So starting in 2010 was when I started training in ancient DNA lab techniques under Hendrik Poinar at McMaster University in Ontario, Canada. I worked on some mammoth samples and some mastodon samples to learn how ancient DNA was extracted at the time and sequenced. And those were some of the very earliest days of sequencing whole ancient genomes. It was so early that there had only been, I think, one or two publications showing that it was possible to get large amounts of DNA from ancient specimens and historic specimens from museum collections. So when I reached out to museums to get passenger pigeon samples to work on, a lot of them said no because they didn't believe that there was very good DNA and they wanted to wait to see if field progressed more. And it was the Chicago Field Museum of Natural History that gave me my first break to work with passenger pigeon DNA. And I extracted that DNA at Hendrik's lab. I raised a small amount of money from family and friends to do the sequencing because it was not a part of my graduate studies at the time. And I got my first kind of real whole genome sequences. And that was right around the time that Ryan Phelan and Stuart Brand were founding Revive and Restore around the concept of the passenger pigeon project to push conservation and biotech farther than anything before. And I got connected with them through a mutual contact, Joel Greenberg, who wrote this book at that time about passenger pigeons and their history. Joel wrote Feathered River Across the Sky. And that's why he was invited to the very first meeting at Harvard that happened in 2012 with George Church and Beth Shapiro, Scott Edwards, a host of incredible scientists. And he emailed me and said, "Hey, these people want to bring back passenger pigeons, and that's something you've always wanted to do since you were a kid, and you've got DNA from passenger pigeons. You should get in touch with them." And one email led to another, led to a phone call, led to the job I have now, which has grown far beyond passenger pigeons into a host of projects around the world for coral reefs, kelp forests, endangered black-footed ferret, Przewalski's horses, and I mean, you name it. We have projects creating genome sequences or innovative technologies for approximately 120 endangered species that occupy every region of the world.
Right. How cool is that? Ben, I want to just digress, before we kind of get into the biotech conservation. Since you've been involved in extracting ancient DNA, now biotech is accelerating. It's accelerating like really, really fast. Because of gene editing technology, CRISPR-Cas9. Recently, there was in Siberia, they kind of extracted the DNA of a zombie. Just wanted your thoughts on that. What could be the implications of reviving something like that?
Yeah, so it's very interesting. Something to know about ancient DNA, which affects, you know, what can really be done as far as de-extinction goes is, of course, if you want to try to recreate an extinct species, you need to be able to sequence its DNA and understand its genetic code in order to make those mutations in a living species and recreate the traits of that extinct species. So without the DNA, you can't even get started. And DNA does not do very well in hot and wet environments. So it's very hard to get old DNA from tropical regions. The older DNA comes from things like caves, where the temperature has been stable and low for a very long time, or from Siberian or Canadian permafrost, where it's been locked in the ground frozen for a very, very long time. Some incredible things have come from permafrost in the recent 15 years. People have revived bacteria from thousands of years ago that were still viable to grow. They've revived old bacterial viruses. So, not viruses that infect people, but viruses that infect those bacteria. And Russian scientists many years ago actually managed to take a piece of plant tissue that was found, culture cells, and grow an entire plant from a plant that was 30,000 years old. Bacteria and plants and viruses have different cellular and protein coat structures than animals. So that helps protect and keep the DNA bound together in ways that it can remain viable for hundreds or thousands of years. Animals, it's much, much harder to get long-term preservation. So when people dig up a mummy of a mammoth or a woolly rhino or a cave lion pup, the DNA is very degraded, and has fallen apart. There's really no majorly intact pieces of DNA left. So it's a complex thing to try to get good material out of. And so I haven't heard of anything new out of Siberia, but it doesn't shock me that someone could be reviving DNA molecules and getting them to grow and replicate in a petri dish because they've done it before with those other microorganisms and things.
Right, yeah, so I think we cusp at such a fantastic point in time where we are able to do that. And I mean, possibly not just revive, the extinct animals also, but possibly maybe also zombies. I mean, these viruses. What could be the implications of that? And so we're sitting at a point in time which is so exciting as well as scary. So Ben, for the people in my audience who kind of don't know what extinction is, will be great if you could start with maybe explaining de-extinction. Why do we need to bring back extinct species? What value does it add, and why does it matter for conservation today?
Yeah. So de-extinction, it's a misleading term. It implies bringing something back from the dead, which is not possible unless you have truly intact frozen cells. As I said, it's happened with a plant from thousands of years ago, it's happened with bacteria from thousands of years ago. It can't really happen with animals from a long time ago. For animals, you need to be able to take those cells and cryopreserve them in liquid nitrogen to keep them in a state able to then revive later, which is something Revive and Restore has helped do with partners at San Diego Zoo and Fish and Wildlife Service for decades old, a black-footed ferret material and Przewalski's horse material. But when it comes to hundreds or thousands of years, animals are the types of things where you have to get a piece of bone or hair or skin that's been preserved either in a museum or in permafrost or in a cave and sequence the little tiny fragments of DNA and map its genome against a living relative where you have an intact piece of genome, an intact genome from their fresh biomaterial. And because of that very first limitation of having to map these tiny fragments of DNA and the limitations that we cannot just create genomes from scratch, we cannot synthesize whole genomes. The idea of de-extinction is that we can take the mutations of an extinct species and edit them into the genome of a living relative. And so this actually creates a hybrid animal, an animal that's the majority of its DNA is a living relative, but it carries the unique genes of an extinct species that if the job is done right, that new animal, that new hybrid will look and behave like an extinct species so that when it's introduced into nature, it fills the ecological role of that species. So, in short, the process is sequencing DNA from an extinct species and editing the mutations of that extinct species into a living relative to create a functional equivalent to that extinct species, something new that fills the void in nature. So, de extinction is about reversing the effects of extinction, but you cannot resurrect an extinct species in its exact extinct form.
Right, right. So Ben, maybe we'll kind of get into that. Maybe I'll kind of try and push back because there are people who are kind of pushing the forefront of maybe synthetic life. Instead of living relative, they are kind of creating artificial wombs. So maybe we'll get into that. But before that, I mean, you gave the process of maybe the genetic engineering and having a living relative to kind of implant that embryo. Will be great if you could elaborate this process. This entire protocol of I mean, what does it take to like genetically engineer and bring back extinct species? What's the protocol? There's stem cell, that's genetic engineering. There's also cloning. Which will be great if you could I mean, maybe elaborate the pieces.
So the biotechnology toolkit for conservation is very diverse, and it has to be because in order to use new biotech tools like genetic engineering or gene editing, which are kind of two different things, but they both manipulate the DNA code of a species, to do it for something like a coral, a black-footed ferret, or an extinct passenger pigeon, these very different organisms, or a plant. All these species have different biology, different reproductive systems, which requires different innovations. So it's a diverse set of tools that depends on the animal or plant you're working with. The engineering part and editing part is kind of the same for everything. You need to be able to culture the cells of your organism in the lab. So you have to be able to take a piece of tissue and put it into a petri dish and grow that tissue into a culture of one type of cell. So you can take a skin sample from an animal and grow the skin cells, or you can take liver cells or some type of cell, grow it in a petri dish. And that's where you can introduce the genetic engineering technology, whether that's older forms of the technology like transgenesis technologies, things like inserting a virus that then inserts a new piece of DNA or a transposon that inserts a new piece of DNA, versus the newer gene editing technologies, which people are more familiar with now because of the incredible breakthrough of the CRISPR-Cas9 system. So CRISPR systems, which are now a diverse tool set too. There's CRISPR-Cas, CRISPR-Cas13, which actually modifies RNA rather than DNA. There's a number of different CRISPRs. There's combined forms of it, prime editing, et cetera. People have been innovating this tool now since 2012, and it can do all kinds of things in a genome. But what makes it special is from the 1970s all the way to about the early 2000s, if you wanted to engineer an organism, one of the few things you could do was insert a new piece of DNA, but you could not direct where that DNA went. So for people that are unfamiliar with DNA, like the human genome, all of the DNA inside a human cell that gives us the traits of who we are is built on long strands of the DNA molecules that are called chromosomes. There's more than one. So a human genome has 23 chromosomes from the mom and 23 of the same chromosomes from the dad, except for the special XY, that if you have X and Y, you're a boy. If you have XX, you're a girl. And with older technologies, your new piece of DNA could go into chromosome 1 or chromosome 2 or chromosome 17. You could not control where it went. You couldn't control if it was going somewhere in the genome where it wouldn't cause any problems, or if it would just go in the middle of another gene and disrupt something. With gene editing technology, scientists are able to design sequences that combine with the editing protein that can direct it anywhere precisely in the genome. So if someone wants to, say, cure a genetic disorder that occurs in a specific gene, they can send the CRISPR system directly to that gene and make the precise change to the DNA code that will turn a disease into a healthy DNA sequence. That's the big promise of this technology. There are a number of human trials in multiple countries that are using this right now to treat human genetic disorders. But people immediately saw that, well, if you can do that, you can do things like engineer plants and livestock to be more climate resilient, or be more efficient at using water and producing food for human beings. So agriculture started adopting this. And then when we formed as Revive and Restore in 2012, was basically, okay, well, if human medicine and agriculture are leveraging and innovating this technology for different ways, we could take this over to help wild species, which have the same problems facing climate change, combating introduced diseases, low genetic diversity and inbreeding problems, as well as the ambitious things we've been talking about of taking mutations from extinct species and putting them into living species. All of that work, that gene editing work, happens in a petri dish with cell culture. You put the technology in there, grow up the cells, you split them into little subpopulations, you sequence their DNA to find out, okay, which of the cells got the right mutations and get rid of the ones that didn't get the right mutations, because it's not a 100% perfect process. But when you get the perfect ones, you grow those up. And the tricky part is then turning those gene-edited cells into an embryo and a living organism. That's where the technology becomes different. If you're working with a mammal, you might use something like cloning, where you take the cell and take an egg cell from a living relative and put the DNA into the nucleus, form the embryo, put it into a surrogate mother, and produce the baby. There are other technologies to use. You can take it that are being innovated that have been done with lab mice. You can take those cells, turn them into stem cells, and then turn those stem cells into sperm and egg, and do in vitro fertilization to make a baby. With plants, it's different. You culture the cells and create things that are called somatic embryos, which can then grow into a bit of a seedling that you then transplant and culture in different ways. In birds, it's very different and very challenging, because birds lay hard-shelled eggs, and so they don't have a pregnancy like a mammal does. So what we do and what we're trying to do for birds for the passenger pigeon project is take the gene-edited cells and work with the reproductive cells called germ cells. So they're called primordial germ cells. Now, we're also looking at whether or not we can create stem cells to then turn them into these germ cells. But basically, you have to use a reproductive styled cell in a bird. And after you gene edit, you implant those cells into a surrogate mother. at the, not necessarily always while it's developing, and a surrogate father at the embryonic stage. So you end up with a male and a female bird that are normal on the outside, but in their reproductive systems, they carry the gene edited cells. And so when they breed, they make the gene edited baby, and the edited baby hatches out. So in the context of the passenger pigeon project, we will take the germ cells of the living band-tailed pigeon, insert mutations, overwrite mutations from the passenger pigeon's DNA into their cells, take those cells, put them into a female domestic pigeon and a male domestic pigeon, grow up those birds, and when they breed, out of their eggs will hatch a new kind of passenger pigeon.
Wow, wow. How cool is that? And how fascinating is this biotech technology? I mean, and the applications is across. You mentioned, it's agriculture, it's de-extinction, coral, then even healthcare. You spoke about gametogenesis. There are people who are trying to kind of figure out, I mean, even single parents, maybe who would want children. I mean, you could do that. So I think we're getting into that like wonderland, but also, I mean, obviously it becomes like scary, like, what are we doing? I mean, are we trying to play God? But I believe that, we humans are the God. I mean, I think we just looking outwards. It's, I think, possibly maybe religion, politics, society, cultures tries to inhibit, the curious lot who are trying to create, innovate. And I think we need scientists, researchers who've got to constantly keep on pushing the boundaries. And I guess that's when we get these crazy innovations which kind of completely changes the world. Yes, obviously, because these tech stack are becoming so and so potential. I think, there should be like, a certain regulation in place so that, that, the when we are kind of experimenting, there's no unintended consequences out there. Now, revive and restore. You spoke about the passenger pigeon project, and you working on this. What's the update on that? And what's the timelines? When do you think, you will get like bring back these great passenger pigeon?
Yes, well, we started the project in 2012, and from 2012 to 2022, it was rather challenging to find funding for the project. It was much easier to find funding to help endangered species. So that part of our organization grew, and the majority of what we do today is genomics and innovation for living species in their ecosystem. The passenger pigeon project and any bird project shares the common barrier of the technology called germline transmission, what I described as in editing those germ cells, putting them into the surrogate parents, and making gene edited babies. That has been done and is still routinely done in the domestic chicken to create birds that have gene sequences for human medical purposes. People are working on trying to create possibly birds that can help improve food systems for human beings. But outside of the chicken, no one has been able to accomplish this technology with another bird. But not a lot of people have tried. So there hasn't been much funding to do anything outside of the chicken because the chicken is the most consumed meat on the planet. And so it's big money, it's a big industry. There's no need to invent this technology for other things where there's lucrative money. So we kind of stepped up and we're like, okay, well, we're never going to create a passenger pigeon unless we get these technologies working for birds other than chickens. And we're never going to be able to then use these technologies for any endangered birds or anything else. So in 2022, I brought together a community of scientists and conservationists that work on birds around the world to form an informal consortium dedicated to kind of moving the science forward. And by bringing that community together, called the International Avian Genetic Rescue Consortium, which now has members from, I believe, about 12 or 13 countries around the world, over 60 institutions, we were able to kind of entice a funder to put in money to advance bird biotechnology. And so since 2022, we have started innovating multiple fronts of bird biotechnology. This work takes time, partly because just getting the lab set up to work with new birds is challenging. People who work with chickens, they have it easy. Chickens can hatch from an incubator and grow up with no parents. 90% of the world's birds, when they hatch, they're blind and helpless. They need parental care. They need a lot of work. Some birds only breed seasonally, so you can only work with their eggs once or a couple times a year, whereas chickens lay eggs every day. So, the chicken is this really great lab animal. Other birds are not. So, just setting up labs to work with other birds is the first big challenging step. We now fund 14 projects around the world in Japan, Korea, Germany, France, and the United States. And they're working with zebra, with not with wild birds right away, but with some non-chickens, things like zebra finches, quail, ducks, parakeets, and of course pigeons, things that will push the science forward beyond the chicken and make it accessible to new species. I'm anticipating some really great breakthroughs in the next 18 months or so. People being able to culture germ cells from other species other than the chicken. One of our labs at Rockefeller University in the last year produced baby zebra finches using this technology. They have a preprint right now on that science, which is really exciting. And we have a team of people creating stem cells for birds. And a new team will start creating stem cells from wild species of birds based at the Mandai Zoo in Singapore. So it's a really exciting time. It's going to be a couple more years before we see really, really cool, groundbreaking stuff. But thanks to that funding in that program, we have been able to now make some advancements for passenger pigeons as well. And so we have at the New York lab at Rockefeller, we have a postdoc sequencing new passenger pigeon genomes, improving the genome sequence of their living band-tailed pigeon, and really delving into the genomics of what makes a passenger pigeon unique ecologically and biologically. And we have teams of people testing new, larger scale gene editing technologies in the chicken as the test case. So at New York University, in Jeff Buca's lab, an incredible young scientist, Anna Berenson, is doing some of the largest scale genome rewriting ever achieved in birds right now, literally as we speak, to take the domestic white leghorn chicken and try to turn it back into its original wild red jungle fowl form. So this is kind of a test for de-extinction. The science for that is not only coming from New York University, it's coming from Dominic Wright and his team from Linköping University in Sweden, a lab that has 20 plus years studying how chickens were domesticated and their genetics. And so we're pushing forward the gene editing to be at the scale we need for de-extinction. We're pushing forward the cell culture and the reproductive technology. The lab at New York has some early pigeon germ cell cultures that appear to be working. Labs in France have early pigeon embryonic stem cells that are growing. And that's awesome. But at the same time, we can't just create the bird. We want to put it back in the wild. And so I've spent over a decade studying the ecology of the species. That was the focus of my master's thesis. And there's still a lot of good science to be done there. So the reason we want to bring back passenger pigeons here in the United States is they played a very, very important role in forest ecology. If people don't know what passenger pigeons are, they were a unique endemic type of pigeon to North America, Eastern North America, that lived in the forests. And they were once the most abundant bird in the world. So in the early 80s, there were anywhere between 3 to 5 billion passenger pigeons. So billion with a B, 9 zeros. And they flocked in flocks of several 100 million to a billion. The largest flock ever recorded may have been 3.7 billion birds in size. And these giant flocks would come into an area of forest, and when they would land on the trees, they would overcrowd on the branches. They would break down tree branches. They would break down entire trees. And as soon as they got settled in after the chaos, they would consume all of the food, the nuts, the berries in the forest, and then they would digest that, and guano would be excreted. Poop would come out all over the forest floor, and the guano would accumulate in inches just over the forest floor. It was incredible amounts of destruction, but that destruction was essential for the rebirth of new life in the forest. So just like a fire or a storm, they were causing the damage to the trees, and the guano was killing everything in the forest floor. and resetting the stage for new life. The guano would break down and become fertilizer, and the broken trees would allow sunlight into the forest floor. And so these birds were constantly perpetuating a new cycle of life for the forest. And when you and by studying the forests of the eastern United States, it's become clear that that constant cycling is something that all of the dominant, once historically dominant species of trees and plants and animals had evolved to be dependent on. They had evolved for that type of ecosystem. Today, we don't have that cycling happening. The ecosystem is shifting greatly. And even though we have more forests today than we had 100 years ago, thanks to conservation efforts as well as changes in agricultural practices, shifting from the eastern United States to the central Great Plains. There's been more forestation, reforestation, but even though we have more forest, it's not undergoing that process. Species of plants, trees, and animals are still declining, even though they have more forest. So our hypothesis that we put forth was if we bring back the passenger pigeon or something that does the same thing in the habitat, a new kind of passenger pigeon, then it will help bring stability and help our forests. But there's not a whole lot of science there other than looking at historic records. So, in addition to developing the new gene editing technologies, the cell culture and those technologies, we are starting an experiment in Wisconsin here in the United States to take pigeon guano from a pigeon farm and dump it all over the forest and break branches, have a team of people go out and do it by hand, and then record what happens to the soil, the chemistry, the microbiology, what happens to the trees, how they grow and respond, what new plants come up and grow after you've put a bunch of pigeon poop all over the ground, and how do animals change and using the habitat. So we will be doing a full-scale kind of ecological study over the next 5 years. This year, we're just figuring out how to do the study. So we're doing a small pilot in the next month where we will be dumping 52 tons of guano. So that's imperial tons. I'd have to figure out what that is in metric. 52 tons of guano over a couple patches of forest. Next year, we hope to then do a larger scale study where we will dump 250 tons of pigeon guano on the forest and study what happens. So we are doing the ecology work to prepare for reintroduction to the wild and understand how this will benefit nature more and do it really, really responsibly and well as doing that science. All of this is happening in parallel and will converge together. So we hope that about in 2029, the cells, the gene editing, the reproduction will all be advanced to the point where we can take the information we're getting from the genome of the passenger pigeon and start gene editing band-tailed pigeon cells and start creating the birds. So if we start in 2029, because it takes a certain amount of time to create the surrogate moms and dads, and it takes time for them to grow up and then breed, we will be looking at creating the first test pigeons sometime around 2032. So sometime in the mid 2030s is when we hope that we have new generations of passenger pigeons hatching. And then we can start the exciting work of trying to breed and repopulate those and then introduce them into the forest in the coming decades.
Well, I think post 2030, I mean, the world is just going to get like really, really exciting as well as scary exciting because, all these tech stack is converging, possibly maybe we might have artificial general intelligence, like a subspecies of sorts, which is as intelligent or beyond, and so cool that, you're doing these experiments and somehow, Ben, I think we are the only species end up destroying things more, everything goes extinct around us, and we've got this natural skill of maybe killing everything, around us, but yes, there are people such as yourself and teams such as yourself who are kind of, working on maybe, creating that balance, I mean, and so really, really exciting. I hope that around 2030, the timelines that you gave, we are able to, I mean, bring back those pigeons, and possibly maybe beyond the pigeons, maybe some animals also. So what's the next step, Ben? Possibly maybe you have a plan around 2030, 2032, trying to bring back this passenger pigeon. From there, would you be taking like a step towards like an animal, reviving an animal? And what the challenge is, the scaling up from like a bird to animal very monumental? What would be the difference?
So mammals are actually a little bit easier to work with than birds since humans are mammals. The majority of knowledge we've gained through human medicine applies a great deal to different types of mammals. So people know more methods of how to grow mammal cells, and there are more options for reproductive biology for mammals. Mammals, I think, is where the science will progress a lot. It's not an extinct species, but for the past 10 years, I've been working to help save the endangered black-footed ferret. The technology, we didn't even have to necessarily innovate a lot of technology because it's just farther advanced for mammals. So when we started working with this species, a species that nearly went extinct, so they disappeared from the wild, an incredible group of conservationists in the late 1980s captured the very last living black-footed ferrets, brought them into a breeding facility, started a program to breed them and reintroduce them. And since 1989, they have bred nearly 12,000 black-footed ferrets over the course of over 30 generations. They're a short-lived animal, so the population today is only about 600 animals, but those 12,000 animals over 30 generations are all descended from just 7 original wild ferrets. So they have a very, very small gene pool, very low genetic diversity, and that could be a threat long term. And this is a threat that we're seeing for lots of species, because over the past decades, well, centuries, and now more increasingly the recent decades, most animal populations on the planet, whether it's fish, birds, amphibians, reptiles, mammals, plants, insects, you name it, most things have experienced huge amounts of habitat loss and major pressure from human beings, and now climate change issues. And so their populations have dropped. And when their populations drop a lot, they lose the genetic diversity that was so important to their natural evolution and their ability to adapt. So we have fundamentally compromised the future of evolution for many species on this planet. But that's something that these biotechnologies can help. So if we can think about bringing back mutations from an extinct species to help an endangered species, to help the ecosystem, we can certainly do the same thing by reaching back in time to specimens from endangered species when they were more diverse, and bring back their diversity into their living populations. So that's something we have started with the black-footed ferret. So fortunately, in the late 1980s, they captured a large number of animals. The only ones that reproduced were descended from 7 individuals, but they managed to get tissue from 2 other individuals that did not reproduce. And they got those tissues when those animals died, sent them to San Diego Zoo. The San Diego Zoo Wildlife Alliance's frozen zoo team created cell lines and froze those in liquid nitrogen. So those cells are in a giant freezer tank full of liquid nitrogen, and they're still alive. They're frozen in time, but they're alive. So in 2014, we helped sequence their genomes and compare them to living ferrets. And they have much more genetic diversity than living ferrets. They're separated not only by 30 generations, but they are unrelated to the founders of those 7 that founded the breeding program. So we proposed that we could clone those cells. A team of scientists led by John Engelhardt at University of Iowa successfully cloned domestic ferrets in 2006. So this was a technology that was already established. This was built after Dolly the sheep was cloned in 1996. A number of species have been cloned. In fact, cloning technology started with amphibians, 2 species of frogs, 1957. It's an old technology that's been done with amphibians, fish, and I think something like nearly 40 species of mammals. In total, about 54-ish different types of animals have been cloned. And so it's a technology we did not need to innovate, and it's one that we could start thinking about using. But no one had ever cloned a wild black-footed ferret. So we had to do a little bit of science along the way that we did with our amazing partners, Viagen Pets and Equine. They're a company based in Texas that does cloning for agriculture and for horses, pets, etc. And so we work with San Diego Zoo, who gave the cells to Viagen under the permission of U.S. Fish and Wildlife Service that were saying, "Hey, help us save these ferrets." And in 2020, they were able to produce a clone from those cells that had been frozen in 1988. And that first clone, her name is Elizabeth Ann. She's just an incredible animal. She's now in her very, very old age. And unfortunately, she had a condition in her uterus that she had to have an ovariohysterectomy. So she did not get to have babies. But we produced 2 more clones in 2023, Noreen and Antonia. And both Noreen and Antonia have had babies. So they were bred with males from the regular population, and their babies are now the first black-footed ferrets since the 1980s that are not just descended from 7 unique individuals, but now 8. And so we've brought a completely new individual into the population from the past. This is the first time cloning has been done to do this. This is the first time a completely new individual from the past has been brought into a population. And so we are restoring lost genetic diversity to this species. Now, one individual is probably not enough. And so we need to look at ways to do more. So we will be looking at how we can sequence DNA from museum specimens of black-footed ferrets and use gene editing to get more of their diversity back into the species. But this is a species that, like many others, is facing a challenge with a new disease on the landscape. So people in Asia will be very, very familiar with the plague. Plague got to North America on a ship from Asia that landed in San Francisco in around the year 1900, and it started to spread through the native rodents of North America, and it got to the prairie dogs in the Great Plains that the black-footed ferrets eat. And so black-footed ferrets have no immunity to this disease whatsoever. So outbreaks of plague kill off the ferrets. They contributed to their decline. It's a major barrier to saving the species today and putting them back in the wild. And because it's something that they never evolved with, and they have now lost so much genetic diversity, it's something they cannot adapt to. We are studying ways to use genetic engineering to give them the immunity they need to overcome that disease. That will be research that goes even beyond our passenger pigeon work, because it's going to take a long time to develop and then test. But we are looking at creating genes that mimic and act like a vaccine. We're trying to look at ways that mutations from the domestic ferret from Eurasia, which is immune to plague, might be able to be brought into the black-footed ferret and make it immune. And this is something that I think is going to become really a major thing for biotechnology and conservation. These are things that we're exploring, understanding for coral reefs and how they're coping with increasing temperatures in the ocean. How can we use gene editing from, say, coral reefs from very hot areas of the world, like the Red Sea, and use their mutations to help coral reefs in the Great Barrier Reef or other places to be able to cope with the ocean heating up. Corals face a lot of disease problems. So are there ways to help engineer them to overcome those new diseases that are happening and spreading more rapidly due to climate change? These types of problems that all animals face with evolving are the things that biotechnology can help overcome. It will never be a complete tool by itself. We can't gene edit a black-footed ferret and then put it in the wild if there's no food for them to eat and there's no wilderness for them to live in. So these technologies work to help conventional conservation work, but it's so imperative that the world's governments and people set aside habitat and start working more on how we produce energy and food and our products in ways that are more sustainable and compatible with ecosystems so that we can live with nature rather than live off of nature. So I think these are all incredible tools, and it's the early days of the science, but it's going to come rapidly. As you said, like the 20s will be an incredible time for this science. I think every decade we're going to be seeing biotechnology doing new and just incredible things.
Yeah, completely. So super exciting. In fact, I was interviewing Andrew Hessel maybe in a couple of weeks back, and he was talking about cloning a human. That's his next project. And you mentioned we've been cloning for the longest time. You start with Dolly, and then especially I think China, it's really big. Cloning animals, because there's a lot of these millionaires who want to bring back their pets. So cloning itself is like really big. And then you were saying with this, the kind of technology stack that you're trying to do, you're making them immune resistant and so on and so forth. And all that help is possibly going to help across maybe the environment also, because I think today we are consuming without conservation. We staring at an existential crisis because the way we treat our environment, because I said in the course of the conversation that we are the only species that has polluted the water that we drink. There's so many wrongs that we have done, but yes, revive and restore. Now there are only few startups in the space which is looking at de-extinction. Besides you guys, there's Colossus Bioscience. That there's not much. Are there any other peers besides you guys who are working on de-extinction? And how do you make this ecosystem sustainable? I was going on the LinkedIn page, and I saw that there's something called as a Catalyst Science Fund and Marine Biobanking and Sequencing Fund. It will be great if you could talk about that too, as well as other peers in the space.
Yeah, so there are more people in the world that are interested in using biotechnology for conservation. There are not very many people really contemplating de-extinction other than Revive and Restore and Colossal. There is Michael Archer's team in Australia trying to bring back the extinct gastric brooding frog. That one's kind of interesting because they don't necessarily have to do a lot of genetic engineering, if any, to recreate that species because they have frozen tissues from that species. So that's a case where they actually get to revive the original animal without trying to hybridize and create a new one. Once they're successful at cloning some of those frozen individuals, to get genetic diversity back, they will have to use some type of genetic engineering to help make a viable population. But that is kind of different because I view that as kind of an extreme recovery of that species. It's actual kind of reviving that species. So it's very different from, say, what Colossal wants to do with the mammoth or what we at Revive and Restore are doing with passenger pigeons. There are teams of people that already beat us to the work. So starting in the 1980s, a group in South Africa started trying to rebreed the extinct quagga, its color pattern. It was an incredible, beautiful species that lived in the area. And they noticed that it was a type of zebra. And they noticed that some living zebras had some traits that were a little bit like quaggas. So they started selectively breeding them. So they didn't have to use biotechnology. They used conventional selective breeding. And today they have some animals that have basically the same traits in color patterning as the extinct quagga, and they've released some of those on some private reserves. So they technically already achieved de-extinction. There are groups in Europe, several different groups that have been trying to use the same breeding concept to recreate the extinct aurochs and release those as part of the Rewilding Europe programs, which are also introducing species that are still found in Asia and Africa that once lived in Europe to get those large species back, like water buffalo, as well as restoring the native European bison. So there's a lot of work in restoration happening around the world. Not a whole lot that happens with biotechnology, but there's a growing movement there, and it's all the same mission. In terms of other biotechnologies, there are teams using biotechnology to help save the endangered American chestnut tree, which was decimated by disease. So they're engineering immunity for that tree. There are groups trying to use biotechnology to help control invasive species. So Island Conservation and their partners at CSIRO and other labs around the world formed the G-Bird group, which is the genetic biocontrol for invasive rodents. And so they're going to be using genetic engineering to try and create male mice that you can release into the environment that will then, when they breed, only produce male offspring. So that over time, over several generations, the entire population becomes dominated by males. And then when there's no more females, then it ceases to continue and we can get rid of invasive left-handed configuration. So yeah, there's a number of academic labs involved in these types of endeavors. So it is a growing space. And we at Revive and Restore help kind of connect all these different communities together. We run a genetic rescue listserv through email. We also host workshops and meetings to connect more biotechnologists to conservation. So we're well connected to a lot of these groups, including the BioRescue group, which gets some funding from Colossal in Europe and Africa, trying to help save the northern white rhinoceros. There are only 2 northern white rhinoceros left in the world, both aging females, but there are frozen cells and frozen sperm and eggs to make embryos. So BioRescue, led by Thomas Hildebrandt, is working to help save that species. They're hoping to do it before the last ones die. But even if the last females die, I am confident they will produce a new generation. They will be using the southern white rhinoceros as the surrogate mother. And to get an idea of who's working in this in the world, there's actually an incredible and beautiful documentary series, a 4-episode series called The Resurrection Quest. It was created by a production team from Mumbai and New Delhi and aired out of Singapore, out of Channel Asia News. And I just had the absolute pleasure of spending 10 days with that group of people filming. And so it talks about passenger pigeon de-extinction, mammoths, northern white rhinoceros. It talks about basically even pet cloning and animal cloning, about how biotechnology and human lives are being changed by this. Beautiful series. I highly recommend people check that out because you'll get a sense of who's working in this in the world. And yeah, and so people are pushing the envelope. We at Revive and Restore are not only hoping to connect all these different people and form this community and create the advocacy for it, but we're always wanting to push the newer technologies. So our newest program, we just funded 4 new projects working with stem cells for wildlife for a couple hundred different species of mammals, endangered and common, for birds, working with germ cells from endangered tortoises. So we're pushing that. We've just launched a project to help try and create methods of cryopreserving amphibian embryos, which has never been accomplished before and would be a game changer for saving amphibians. And we have our advanced coral toolkit, which is creating stem cells from corals, studying their genes, creating probiotic treatments to help them after bleaching events, methods of detecting metabolites in the ocean that can tell us when there's going to be a bleaching event, as well as simply just creating and sequencing more reference genomes and population genomics from species around the world so that that information in the DNA can make better conservation actions. We fund 50 projects currently around the world, led by universities in 21 nations. And yeah, as I think I said earlier, those projects are innovating technologies with laboratory animals, completely new things that have never been done before. But they're also using genomics and other tools that are already available to help about 130 endangered species around the world. And it's a very, we're growing as an organization, and I think we're pushing the cutting edge boundaries. We're hoping to start helping create AI tools to accelerate genomic information, and possibly creating stem cells using AI information to improve the cell culture and the data mining that helps create that. So yeah, if our goal is that if it is an idea that can be formed from biotech that can help species, we will take on the high risk to hopefully create that high reward that other funding agencies like the National Science Foundation or government agencies around the world may be a little more hesitant to step in and help. We're the ones who want to stick our head out there and be at the front.
Paint a picture of the next maybe like 5 to 10 years, because biotech is converging with artificial intelligence. We already have these foundation models, biotech foundation models, not just large language models. We have these biotech foundation models. How is this convergence with AI gonna accelerate the work that you're doing? I had the privilege of interviewing Dr. Hossi Zainer, who's like a biohacker, and she was mentioning that her dream is to kind of create this mythological creature, the dragon. And then obviously, the Jurassic Park, there are lots of people, who kind of, maybe thinking that maybe we could kind of bring back these dinosaurs. What's your thoughts on that? I mean, is there fear? Do you have skepticism on something like that? Do you think even that something like that is possible? Because you mentioned in the course of the conversation that mammals are more easier than avians. So would love your thought on that.
Yeah, so a lot of times when we bring up talking about passenger pigeons or mammoths or things like that, people think of the movie Jurassic Park. They think of dinosaurs. They think of where technology could go. When you bring up AI, we think of movies like Terminator. We think of these things where things go really bad. And while I fully agree with people, there are many concerns, a lot of ethics and regulations to be sorted out that have to happen not after we create these technologies, but as we create these technologies. There are some things that will always be to the realm of imagination. So there is no dinosaur DNA left in the world to sequence. So we cannot understand the genome of, say, a Tyrannosaurus rex or a Triceratops. Without being able to understand their genomes, we can't really recreate them and their traits. Maybe possible to take the living chicken and give it a tail and give it claws and give it teeth and make it look more like a dinosaur from 66 million years ago. But that's just a modified chicken. And we can do that. It's called reverse evolution because we can do that because chickens still develop when they're embryos with a tail and with fingers. They still develop with the traits they had from millions of years ago as theropod dinosaurs. So, that's a pretty big stretch to create a chicken that looks a little bit like a little tiny velociraptor to a stegosaurus. So there are some things that will just be kind of not necessarily always impossible, but some things that, if someone in the future someday wants to create their own real Jurassic Park, they will be kind of creating those species completely from scratch. Once we understand DNA enough to be able to make whatever DNA sequence we want, we may be able to make organisms that have never been made before. And people are doing this in bacteria, where they're changing bacterial genomes at such scales that they're rewriting kind of the code of their life. And there are some things that we've decided as a community probably shouldn't happen. One of the things that's kind of the most concerning, because we can kind of re-engineer bacteria so much, is something called mirror life. So for people that aren't familiar with biochemistry, the molecules that make up our bodies can all come in two forms. So when a DNA molecule can have a right-handed configuration or a left-handed configuration. I have to look up in a book, but I'm pretty sure all the molecules that make up life as we know it are left-handed configuration. We don't know why, but for some odd reason, 3 billion years ago when life formed on this planet, it formed in a way where every molecule worked with its left-handed form. And because DNA, the way every molecule in a cell works, it interacts based on shape. You cannot introduce a right-handed molecule. It won't fit with the system. But right-handed versions of DNA molecules and proteins and other things exist in nature. They can't produce life the way they are right now, but they exist. And so people have figured out that you can synthesize right-handed versions of molecules, of living molecules. And they call it mirror life. And we're getting to a point where we may be able to create self-replicating simple cells that are made completely of molecules that are not recognizable by our bodies. I was at a meeting of scientists from around the world in February, and it was basically determined that this is a science that is so astronomically dangerous that it just should not be done. Because imagine someone creates a virus that cannot be recognized by the human immune system at all. Theoretically, it should not make us sick, but we don't know how it would actually interact with our body. All we know is that our immune system would be unable to recognize and conform to it. We don't know if we consumed a food made from mirror life molecules, if we'd be able to digest it. or what would happen. The thing is we literally just don't have any idea. And if those are replicating things, they'll form their own ecosystems that possibly do all kinds of things. So it was decided, and people signed a treaty actually that was, I think, recently published. It was decided that there is something to learn from creating these molecules in a laboratory and studying them, as long as those molecules cannot replicate. But if anyone creates a self-replicating mirror system, that is an avenue of research that should never be pursued because it is simply just so unpredictable. It's the kind of thing that not even AI could help us understand because there's just no data to create there. Maybe someone on another planet someday could study this type of thing, but on Earth it is just not safe. But this brings up AI. How safe is AI? In biology, people are already using AI to study genomes. The human genome is 3.3 billion letters of code. There are trees out there that have 30 billion letters of code. There's a fish that has 90 billion letters of code in its genome. Genomes are huge amounts of information, and crunching big amounts of information is the thing that machine learning and AI was basically developed to do. So there's a lot in biology that could be accelerated using AI to try and understand and predict what genes are responsible for what traits and what genes interact. So there's a huge revolution coming in AI in that space that will help accelerate human medicine, agriculture, as well as our conservation work. But to turn genetic information into something that matters and can be used, as I said, you have to be able to use it in cells, living cells, in a petri dish. And growing cells from organisms is incredibly difficult to do. Cells do not like living outside of an organism's body. They don't like living in an artificial environment. Keeping them happy and growing them is challenging. It requires a lot of nutrients and understanding. Turning one type of cell into a stem cell and then those stem cells into other type of cells requires knowledge of the genetics of those cells as well as how to keep them alive and growing and changing. So it's a 3D, it's no longer just a DNA information, it's a 3D interactive environment through time. And so AI can certainly help there as well, because today, people who have just spent decades looking under a microscope at cells know how to do that work, but it's very artisanal. It's not very quantitative. To get someone else who can grow cells really well requires years of training from one person to another. It's really an apprenticeship, artisanal science. It's not a replicatable kind of true hard science the way we want it to be. So we want to accelerate this type of science. So AI can help with that in taking images of cells as they grow, in starting to learn, okay, what are the traits visually as well as from genetic information that tell us, oh, this cell is becoming a stem cell. It needs new growth factors. When does it need a new media change? So there's a lot that AI will do in terms of cell culture, in standardizing and accelerating that science, in understanding genetics. And there's a lot AI is doing right now in helping us understand landscapes, using satellite imagery and other types of data combined to figure out what are the environments that could still be healthy enough for species to live. It's helping people go out and find species that are so rare we haven't been able to find them in regular monitoring. And it's helping identify environments to reintroduce species. It's helping figure out which environments might be susceptible to certain types of climate change. So, in terms of biological science, AI is going to be an absolutely revolutionary and vital tool. But once again, every new tool can be used in ways that are responsible and good, as well as ways that could create problems. So I do hope sensible regulation comes in for AI, particularly in how it's used in social discourse and politics, etc. I would like to live in a world where we can tell the difference between reality and invented stories and lies. And that's important across everything. So in the sciences, it's going to be important to know that the information being given to people is truth.
Right. Right.
So in the past, before there was even AI, there were people that faked scientific data. Notably the man that's published the paper that said vaccines cause autism. That data was fake. And it caused decades and billions of dollars of wasted research and now thousands of human lives because people believed a lie. Well, how are we going to tell the difference between fake data and real data if AI creates fake data that's so believable? So, we face these issues, I think, in being genuine and transparent in our science and what we're doing. And this is where it really comes down to the fact that science is done by people. People are flawed. But if we're genuine and honest and try to make human connections as a society, we can work together to do these things in a way that benefits humans and nature and can be a good thing for the world in the future.
Like a last question. Yes, I think we're sitting in a fantastic point of time. We have democratized knowledge through these large language models, and we are at the cusp of possibly democratizing DNA itself. If we have like a model that kind of is able to understand the genome code, and maybe give that access to anyone and everyone. I mean, what the world would look like is obviously exciting as well as scary. My last question, I mean, what comes, maybe paint a picture like in the next 10 years, think that we're sitting in 2035, what would revive and restore, I mean, look forward to, achieving at that point of time. And the maybe in the course of the conversation, I was kind of pushing, saying, I mean, I can want to kind of push back on designing life, obviously not like the mirror life kind, which could be like really, really harmful. Designing artificial life, and because we're at the cusp of possibly tinkering around, these genome code and possibly creating life itself, and also artificial womb, because you spoke about that at this point in time you need a living relative, but there are people who are working on artificial wombs. So three questions, like the revive and restore by 2035, artificial life and artificial womb.
Yeah. So, I mean, artificial life in terms of microbes, hello, hey, are already coming. As I said, people are engineering genomes of microbes in incredible ways and basically creating DNA codes, configurations that are suited towards particular needs. So, such as like trying to break down plastics or break down oil. So, I think we are in the decade that, thanks to AI as well, which is now basically inventing new DNA sequences to test in these microbial systems. We are in the decade where we will confront the possibility of releasing bacteria and other microbiology systems into environments that do jobs for human beings that we could not do in any other way, jobs that could be safer. So, I mean, trying to clean up pollution and toxins is very challenging. You usually, sometimes you have to use other toxins to try and create chemical reactions that bring those things out of the system. But these living systems could do it better. And there are, of course, potential concerns with those artificial life forms then becoming invasive species. We, it's no one's done it yet, so we have time to kind of figure that out. But there will be a time when we're using these tools, if not in nature, in human factories and environments to do new things for us. So I think there's a very promising future for artificial life at the microbial level. When it will come to, synthetic or artificial plants or animals, there will be a day when human beings can do absolutely incredible things. You look at the writings of Jules Verne in the 80s, and the things he wrote were not possible at the time. The things H.G. Wells wrote about were not possible at the time, but things that they wrote and envisioned became reality decades later. So things that seem like science fiction today could be real in the future. Could we 3D print entire life forms in adult phases of their life and just turn them on and give them consciousness somehow? Could we create organisms that have never existed in any means? Could these organisms be created to help colonize Mars, asteroids in the asteroid belt, moons of Jupiter and Saturn? Could we create interstellar travel and create systems that help create ecosystems on distant planets of distant stars? I think these things are all in humanity's future. they're not happening so fast that we're going to have to be the ones dealing with a lot of those ramifications. But right now, there will be applications of certain artificial life forms at microbial scales that we have to deal with. The more we can 3D print organs and create artificial systems, we will get to a point of, say, an artificial womb, which can help, say, give birth to a de-extinct mammoth or a giant ground sloth. There are these species that are extinct that are hundreds of times larger than their living relatives, and the ground sloths are a great example. Ground sloths could play a role in today's nature. The plants they once ate and helped disperse seeds of are still alive. And we need our large species back, what we call the megafauna, the super megafauna, things the size of elephants and rhinos, used to live everywhere in the world. And human beings, those are the first types of animals that human beings wiped out as they spread across the globe. And they played a unique role in ecosystems. And those are things that we should work to restore, but we can't create a giant ground sloth without an artificial uterus because the baby is larger than its living relative. But if you can create an artificial womb, now you're creating the ability to allow people to have babies in a completely new way, which can sometimes seem scary. Can also, right now there are people who cannot have children who use surrogate mothers. Well, their main, and just like you wait for an organ donor on an organ list, artificial organs, 3D printed organs will help that. Same thing, like when a woman cannot have children and she's waiting for a surrogate mother, there might not be a willing mother that wants to have her baby. Being a surrogate mother is emotionally challenging. It's very difficult to grow a baby inside of you. We as men will never understand. But to grow a baby and then give it up, knowing that it's not yours for someone else, that's an incredible, magnanimous feat of humanity. An artificial womb opens up a new industry for people with fertility problems. It opens up the possibility that if a mother in her third trimester of pregnancy is in a car crash and the womb is pierced, that you can save the baby, take it out of her womb and finish the pregnancy in an artificial womb, save the baby and the mother. These things will open up a lot of incredible things for human beings. The stem cell technologies we're talking about will enable same-sex couples to have their own genetic children, two fathers to have a baby together, two mothers to have a baby together, women that cannot have babies to have their own babies, men that can't have babies to have their own babies. So, there's an incredible new world coming, and a lot of people might think it's scary. But I think we can come back to when in vitro fertilization was first used. A lot of people had the same fears about that. Is this a baby in a test tube? Is it an artificial baby? No, it's a real human being. And tens of thousands of parents around the world have had babies through in vitro fertilization. And there are probably plenty of people listening to this podcast right now who are born from in vitro fertilization. So there's really not a whole lot of difference in the future if there's a human being walking around that was born from stem cells from two moms or stem cells from two dads, or a baby that was born from an artificial uterus rather than a surrogate mom. As long as we can create these technologies in a way that produce healthy people, healthy animals, healthy plants, then these are going to be someday just part of everyday life in the world. And hopefully in a way that creates that sustainability and coexistence with natural ecosystems and natural resources so that we can feel a little less crowded on this planet. We can feel less starved. We can feel less sick all the time. We can have clearer skies and see better stars and better sunsets. The world is in a bad position right now with climate change, pollution, and all of the political problems we have. And there are a lot of those problems that can be solved simply by changing infrastructure and government and how we do things. But biotechnologies in agriculture and human medicine and conservation can step in and help complete the picture so that we can do even better things in the future, for that future of just kind of, utopian coexistence. I'm tired of thinking about the future as a dystopian post-apocalyptic world where we never figure out how to save the world and help human beings at the same time. I think the future of, say, like Star Trek, I'm a big Star Trek fan, where people get it all figured out and we actually are healthier and happier and everything works. That is a future that is still accomplishable. Even though it might seem like there's no hope, there's always hope for that future as long as we keep building for it.
Thank you so much, Ben, for taking time and being part of the podcast. You painted such a beautiful picture. And I guess, in optimism, there's the hope, there's always like a brighter future. And would love to end with this last note. I love this song by this legend John Lennon, Imagine. Imagine like a better world. And thank you for taking time and being part of the podcast. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you guys. Bye. Thank you, Ben. Really appreciate this.
Thank you so much.